Hello. We at Pharmabase, as part of our commitment to animal health and in partnership with Embrapa Swine and Poultry, have created a series of informative videos covering techniques for necropsy and sample collection in swine. The correct diagnosis of swine diseases is both a first step and one of the most important steps for decision-making in disease control. Therefore, we have invited a pathologist who is also a specialist in swine health to present this specialized technical content in a practical manner so that you as a veterinarian can be more assertive in diagnosing swine diseases. In this video series, Dr. Marcos Moraes from Embrapa Swine and Poultry will share with us his best practices from animal selection to careful evaluation of the different internal systems. Dr. Marcos, thank you for sharing this knowledge with us. The floor is yours. I thank you, Pharmabase, for the invitation. Our subject today will be the use of laboratory diagnosis in swine production. We will talk about how to properly plan the laboratory diagnosis of a clinical profile. In many situations, the veterinarian may arrive at the diagnosis by means of a good clinical analysis or combine that analysis with a necropsy of one or more dead or sick animals. However, in other situations, due to the similarity of the clinical symptoms and lesions produced by many various diseases, it may be necessary to seek laboratory assistance in order to arrive at the definitive diagnosis of the cause of the problem. Firstly, it is important to choose a reliable laboratory based primarily on its track record, the tests it provides, and the technical staff of the laboratory. Maintaining a good relationship with the veterinarians in the laboratory is extremely important, especially when requesting clarifications regarding the sending of samples, the availability of tests, assistance in interpreting test results, and other questions that may arise. It is important that the clinical veterinarian be aware of the fact that they have ultimate responsibility for providing a final diagnosis, and that the laboratory will only provide test results, which are tools to assist in this diagnosis. The clinician will use these test results along with medical records, responses to possible treatments previously administered, and the evolution of the clinical profile to arrive at a definitive diagnosis. One of the most critical points for success in laboratory diagnosis is the choice of the animal or animals from which to collect the samples. Those who work with diagnosis know that this is the point at which most failures occur in the diagnostic processes. If there is an error in animal selection, the entire subsequent process is compromised. As for the number of animals to collect samples from, this will primarily depend on the stage of production and the size of the farm, as it involves costs. For animals in the farrowing and weaning stages, it is generally recommended to select between 2 and 4 piglets, which sufficiently represent the clinical profile to be diagnosed. In larger animals, it is possible to work with a smaller number. It is necessary to select those animals that best represent the clinical condition to be diagnosed. At this point, the most common mistake is to select emaciated animals, where most of the time the cause of the emaciation is different from that of the animals with the clinical condition to be diagnosed. The selected animals should be in an acute phase of the clinical condition, i.e., on the first or second day after symptoms appear. Selecting animals in a chronic phase makes it difficult to identify the primary cause of a condition, because at this stage, typically, secondary or opportunistic infections are usually most prominent. One other problem is that some pathogens do not remain in tissues for very long, so in chronic phase animals, it may no longer be possible to isolate or identify those pathogens. This factor is very important for influenza and rotaviruses, for example. In the case of respiratory diseases, particularly influenza, the use of a thermometer can help in the identification of animals in an acute phase. Animals with clinical symptoms, rectal temperature above 40 degrees Celsius, and still in good physical condition are ideal for sample collection. Even when the objective is the diagnosis of a clinical profile of emaciation or wasting, it is still necessary to select animals at the initial stages of the disease for sample collection. Animals that have been injected with antimicrobial medication should not be selected, as this medication may impair or prevent the isolation of any possible bacteria involved. The collection of samples from animals found dead is not recommended unless it is certain that death occurred less than 2 hours previous. For a proper complete necropsy, including the collection of samples for laboratory tests, necessary materials include autopsy gloves, lab coat and boots for the veterinarian's protection, equipment for stunning the animals, knife and honing steel, bearing in mind that a good, well-sharpened knife greatly assists the necropsy, surgical scissors and forceps, a source of flame, which can be a Bunsen burner or a cotton ball soaked in 70% alcohol for the disinfection of the instruments, a saw for opening the cranial box and cutting other bones, sterile syringes and needles, swabs, and new plastic bags for packaging samples for microbiological analysis, wide-mouthed vials, and 10% formalin, preferably buffered, for sample fixation for histopathological and immunohistochemical tests. Buffered formalin is usually provided by the laboratory. For necropsies, it is necessary to first euthanize the animal. There are several methods for euthanizing swine. In order for a method to be considered humane and acceptable, it must be swift, effective, and reliable, inducing a rapid loss of consciousness and death, thus minimizing the pain and suffering of the animal. In light of these considerations, electrocution has been the method most commonly used by farmers to euthanize swine with unfavorable prognoses or for the collection of samples. There are several models of equipment for the electrocution of pigs, most of which were developed for abattoirs, and which are excessively complex and expensive for euthanizing animals in farms. However, electricians can assemble simpler models that are safe for this purpose. The basic equipment consists of a cable with 2 parallel jacketed wires containing 2 electrodes in an alligator clip at one end, an interrupter system for safety in the middle portion, and a plug at the other end. One of the primary factors for the safety and efficacy of the electrocution process is containment of the animal. For euthanizing pigs by electrocution, the electric current must pass simultaneously through the brain and heart. To perform this procedure, one of the electrodes is secured to the upper edge of the animal's ear, near the base, and the second electrode is secured to a location posterior to the heart, which may be the skin of the abdominal region, the base of the tail, or the groin fold. After correctly fastening the electrodes on the animal, plug the cable into the electrical outlet, keep away from the animal, and switch the current on. The current should remain on for 15 seconds. After that time of electrocution has elapsed, the switch is turned off and the animal is bled. It is always recommended to bleed the animal before the necropsy because the less blood that is present at necropsy, the easier it is to note any alterations. In the next videos of this series, we will discuss the different stages of the necropsy process.